Lecture 4, we'll review the 5 key questions from evolutionary cognitive science. We'll start with an in-depth overview of one of the 5 questions, the question of ontogeny, and we'll consider the example of the development of fear of heights. We'll end with a broad overview of the remaining 4 questions, the questions of phylogeny, proximate and ultimate function, and the question of hardware. To appreciate these 5 questions, we'll apply each question to the psychology of fear. We'll begin with the study of ontogeny or development by looking at the development of fear of heights. Next, we'll turn to the question of phylogeny or evolution and look at phobias as evolved biologically prepared fears. We'll then look at the question of proximate functions by examining xenophobia as fear of outgroups, and then we'll turn to ultimate functions by looking at fear of outgroups as a pathogen prevention mechanism. Finally, we'll look at the question of hardware and consider the biology of fear and chemical treatments of anxiety disorders. We'll start by looking at the question of ontogeny, or the question of development. Ontogeny refers to the course of development, especially of an individual organism. Compared to many species, human infants are born in a relatively underdeveloped state. Moreover, resources for development are precious and scarce. Devoting resources to develop one trait prematurely might preclude these resources from being used later for another trait. While the development of the infant's heart and lungs is crucial for its survival, the development of its capacity for language is not quite so urgent. Thus, it's not surprising to observe in a preterm or premature infant that their early development prioritizes developing vital bodily organs at the expense of cognitive development. And while a delay in cognitive development, such as a delay in language acquisition, might put the child at an early disadvantage, this is a disadvantage from which the child can easily recover, whereas a delay in the development of the child's heart and lungs or other vital organs could have serious repercussions for the child's survival. Let's now turn to the question of development of fear of heights and ask, when is a fear of heights useful? Is the development of fear of heights something that is crucial for survival for a human infant? Or is it something that can develop later on, like the capacity for language? Human infants typically cannot locomote on their own until about 6 to 10 months of age. Thus, it's not clear what use a fear of heights would be in an infant that can't crawl. This simple observation about the delayed onset of self-propelled locomotion in human infants has led developmental psychologists like Joseph Campos to study the development of fear of heights in human infants. Campos designed a series of clever experiments modeled after early research on fear of heights in other species such as cats. Many of these studies were done with an apparatus known as the visual cliff, in which psychologists constructed a laboratory environment consisting of a checkerboard cliff covered with a large sheet of plexiglass. In this image, the child is placed on the shallow side of the visual cliff, while to the left the experimenter observes, while the mother on the deep side of the visual cliff beckons the child to come near. Although the visual cliff looks like an actual cliff, the child cannot possibly fall down into the deep side because it is covered with a large sheet of clear plexiglass. In a classic study of the development of fear of heights in kittens, experimenters Held and Hines used the visual cliff and another unique apparatus known as the kitten carousel. In this apparatus, one kitten is secured inside of a box where it's not able to move, but it's able to observe the surrounding environment. The box is connected to a long pole. At the other end of the pole, a second kitten is connected to a harness. The second kitten is not only able to observe the surroundings, but can move. Because the harness securing the second kitten is connected to the pole, any movement by the second kitten causes the other kitten secured inside the box to also move. Held and Hine later tested these kittens on the visual cliff. Interestingly, kittens that were held passively inside the box inside the kitty carousel displayed no wariness of heights. However, kittens in the harness, whose active movement controlled the kittens in the passive box, these kittens who actively moved in the kitty carousel displayed a clear wariness of heights when exposed to the visual cliff. Inspired by these findings, Joseph Campos conducted a series of studies with the visual cliff to look at the development of fear of heights in human infants. Campos wanted to know whether self-propelled locomotion, whether in a kitten or in a human infant, serves as a developmental trigger activating fear of heights. Held and Hine studies had used the kitten's reluctance to walk across the visual cliff as an indicator of the kitten's fear of heights. Another important feature of the original Heldenheim study was that they experimentally controlled whether the kittens had any experience with walking. By contrast, the kittens in the harness had much experience with walking. Joseph Campos faced a number of challenges when he attempted to apply Heldenheim studies of kittens, a study of the development of fear of heights in human infants. Campos knew that it would not be ethical to control a human infant's experience with walking, so he decided to examine a group of 7-month-old infants. It's not uncommon to observe that some 7-month-olds can walk or crawl, and others can't. In this manner, Campos divided his group of 7-month-old infants into 2 groups. The pre-locomotor group, consisting of infants that cannot yet walk or crawl, and the locomotor group, the group of infants that were already crawling or walking on their own power. To assess fearfulness in these infants, Campos measured their heart rates as he lowered them safely over the visual cliff. Prior work had shown consistently that heart rate decelerates in infants who are in a state of non-fearful attentiveness, but accelerates when infants are showing a defensive response. Finally, Campos randomly assigned infants to a walker condition or a control condition. Although the walker was of little use to the locomotor infants, they were already walking. For the pre-locomotor infants, by contrast, the walker provided them with an artificial experience of locomotion. The walker manipulation allowed Campos to create 4 experimental conditions. 2 of these conditions occurred among the pre-locomotor infants, who were randomly divided into those provided with walkers and a set of controls, consisting of pre-locomotor infants without a walker. The other 2 experimental conditions consisted of locomotor infants randomly assigned to either a walker condition or a control condition where they had no walker. Locomotor infants in the walker and control condition have already had natural experiences of self-propelled locomotion. Thus, it's not surprising that both groups of locomotor infants show heart rate increases in the visual cliff experiment. Thus, the key comparison is between the pre-locomotor infants given walker experience and the pre-locomotor infants in the control condition. This comparison is important because the pre-locomotor infants in the control condition have no experience with self-propelled locomotion, whereas the pre-locomotor infants in the walker condition have been given an artificial experience of self-propelled locomotion. Consistent with the idea that the onset of self-propelled locomotion serves as an important developmental trigger of fear of heights, Campos found that only those pre-locomotor infants given the artificial walking experience showed an acceleration of heart rate indicative of fear of heights. Having looked in depth at the question of ontogeny or development, we'll now turn to a broad overview of the 4 remaining key questions of evolutionary cognitive science. Continuing with our application of these 5 key questions to the psychology of fear, we'll turn now to the question of hardware and briefly look at the biology of fear and chemical treatments of anxiety disorders. When we look at the physical basis of fear and anxiety disorders, these physical descriptions can range from the macro level of brain and neural circuitry to the micro level of molecules and synapses. We'll first take a quick look at some of the brain circuitry underlying fear. Neuroscientist Joseph Ledoux argues that there are 2 brain pathways to fear. One direct route goes from the thalamus to the amygdala to produce an emotional response. Another, more indirect route passes through the neocortex and the hippocampus before producing an emotional response. According to Ledoux, the advantages of having a direct and indirect pathway to the amygdala is flexibility in responses. When danger is perceived, the fast and direct pathway is useful because it saves time. The longer pathway is useful because they allow for a more thorough evaluation of a situation that can help people to avoid inappropriate responses to certain situations. A description of the physical basis of fear might include not only descriptions of brain circuitry, but also descriptions of brain chemistry. The brain and nervous system are made up of cells called neurons, which communicate with one another using neurotransmitters such as serotonin. Neurotransmitters like serotonin are secreted by one neuron into a cell junction called a synapse. There, the serotonin binds to structures called receptors attached to a second neuron. The receptors allow the adjacent neuron to take up serotonin, leading to chemical changes in other parts of that cell that regulate brain function. One of the issues that many people with anxiety have in common is that they tend to suffer from low levels of the neurotransmitter serotonin. Prozac belongs to the selective serotonin reuptake inhibitor, or SSRI class of drugs. Prozac is mainly used for the treatment of major depression and anxiety disorders. It works by blocking the absorption of the neurotransmitter serotonin in the brain. We'll turn now to the question of phylogeny and examine how phobias can be considered biologically prepared evolved fears. It has long been known that many adult primate species exhibit a marked fear of snakes. The question of phylogeny might ask whether fear of snakes is an evolved adaptation or whether it's a byproduct of a more general social learning mechanism that allows a primate to learn to fear any object that it observes another member of its species displaying fear towards. In a study of rhesus macaques, Susan Meinecke examined whether fear of snakes is a biologically prepared fear response. In this study, monkeys were placed in a large plexiglass box, and they were trained to reach rapidly across the plexiglass box to obtain a desired food treat such as a raisin. Once training had established a baseline of rapid reaching for food, the monkeys' reactions to different test stimuli in the plexiglass box were tested. The test stimuli included real and toy snakes and a neutral object, a flower. Meinecke then did something very interesting. She edited videotapes showing wild monkeys displaying fear of snakes. She then created 2 videotapes. In one videotape, the monkey was depicted displaying fear of an actual snake. She edited the second videotape to make it appear as if the monkey were fearing a flower. The naive rhesus macaques in her study were then randomly assigned to view one of the 2 videotapes. In a pretest, Meinecke found that naive monkeys that had never been exposed to a snake or a flower reached very quickly for the food despite the presence of the snake or the flower. However, consistent with the idea that fear of snakes is an evolved, biologically prepared fear, in the post-test, after the naive monkeys had been exposed to one of the 2 types of videos, Meinecke observed that those monkeys who watched a video of a member of their species showing fear to a flower still reached very quickly for the food in the presence of a flower. However, consistent with the idea that throughout evolutionary history, snakes have presented a consistent danger for monkeys, whereas flowers have not, Meinecke observed that exposure to the video showing a member of their species exhibiting fear to a snake resulted in an increased wariness to reach for the food in the presence of a snake or a toy snake, but not in the presence of a flower. At a post-test several weeks later, these same monkeys were still exhibiting an increased latency to reach for the food when a snake was present, but they showed no such response when a flower was present. These findings seem to suggest a special biological preparedness for learning a fear of snakes, but no such preparedness for learning fear of flowers. We turn now to proximate and ultimate functions and look at xenophobia, or fear of strangers, as a pathogen prevention mechanism. Xenophobia refers to a fear or contempt of foreigners or strangers. It's related to ethnocentrism, which is the tendency to look at the world primarily from the perspective of one's own culture. Ethnocentrism often entails the belief that one's own ethnic group is the most important and that some aspects of its culture are superior to those of others. When applied to xenophobia, the question of proximate functions refers to what happens when xenophobia has been activated and what activates xenophobia, whereas the question of ultimate functions refers to how could xenophobia enhance survival or reproduction. Is xenophobia an adaptation, a byproduct of an adaptation, or just noise in a very complicated psychology? To look at what happens when xenophobia has been activated, let's consider the case of Mary Shelley. Mary Shelley was an English novelist, best known for her Gothic novel Frankenstein. In the early 19th century, while traveling to Switzerland via France and Germany, Mary Shelley made the following comments about the French and the Germans. Regarding the French, Mary Shelley wrote in her diary, "Our companions in this voyage are the meanest class, smoked prodigiously, and were exceedingly disgusting. The horrid and slimy faces of our companions in voyage, our only wish was to absolutely annihilate such uncleanly animals. 'Twere easier for God to make entirely new men than attempt to purify such monsters as these." About the Germans, she wrote, "They were squalid with dirt, their countenances expressing everything that is disgusting and stupid." At a proximate level, ethnocentrism and wariness of strangers is often expressed in terms of a reaction to them as being disgusting and dirty. To explore the possible ultimate functions of these symptoms of xenophobia and ethnocentrism, we turn now to a study by Carlos Navarrete. In this study of pregnant women, Navarrete looked at women in the first trimester of pregnancy. These are women who are more vulnerable to disease than women in the second and third trimesters. Consistent with the idea that some of the symptoms of xenophobia may have arisen evolutionarily as a pathogen prevention mechanism, Navarrete found that pregnant women in the first trimester not only rejected more food and experienced more nausea, but they also showed more intergroup bias and ethnocentrism. Returning to our example of Mary Shelley, her rather xenophobic comments in her diary seem at odds with her liberal and progressive character. Yet, consistent with some evolutionary psychologists' views on the ultimate causes of xenophobia, you may not be surprised to learn that Mary Shelley was in the first trimester of her pregnancy when she wrote these xenophobic comments in her diary.